Manish, what about drug development outside of the holy grail immunotherapy? Certainly we're looking at other things. We talked about other targeted agents. Talk to us about some of these FGFR agents, what's the stemness inhibitor?
Right. So the BBI-608 is a stem cell inhibitor, or stemness phenotype inhibitor. And we examined that in gastric cancer in the second-line setting, and unfortunately that was a negative study. The mechanism of action is actually kind of curious. It targets STAT3, which actually affects stem cells, but it also affects the tumor microenvironment. So there was a hope of that actually augmenting immunotherapy or working like that. So we actually also did a phase 2 study of the drug BBI-608 with immunotherapy. We haven't seen much effect of that. Another drug that was actually a negative study was the MMP9 inhibitor, which also targets the immune microenvironment, and that was andiluximab, which is from Gilead. And that was a first-line study, and we also combined that drug with immunotherapy in the second-line setting, and we're doing a lot of pharmacodynamic markers to see how we can understand the immune microenvironment.
And what about FGFR inhibitors and FGFR translocations, or I shouldn't say translocations, but fusions, yeah.
Yeah. So 5' has a very nice drug that targets FGFR3, and that study has shown some activity in these tumors that are overexpressing FGFR3. We actually participated in that study, and although in the advanced setting, the expression of FGFR3 is not as prevalent, but in earlier line settings it can be quite high, 20–30%. So they've actually embarked on a phase 3 study examining that.
And so the other thing that was big news at ASCO was a plenary session looking at patients with BRCA mutations for pancreas cancer, right, looking at the use of PARP inhibitors. So, certainly this DDR phenotype is being evaluated even in gastric cancer. David, something that people have looked at or are continuing to look at. Tell us a little bit more about these patients.
I think it's a work in progress. Yeah. We sometimes have just somatic BRCA mutations. And from our experience, you really have to have loss of heterozygosity to see platinum sensitivity. So just having a somatic BRCA mutation in isolation may not convey that sensitivity to platinum agents. The PARP inhibitor experience was a little bit disappointing. The trial combining paclitaxel with olaparib versus paclitaxel alone, even in a potential biomarker-enriched population, didn't show a clear survival benefit. There was patients that were ATM deficient, might do better with PARP inhibitors, but even with an enriched population, the trial was negative. There was a trend. They set the bar very high in that trial for a large improvement, and also the ATM population was relatively small in that trial. But not a clear signal yet for PARP inhibitors in esophagogastric cancer, but ongoing trials looking at combinations of these agents with checkpoint inhibitors and TKIs to see whether we can build on what looks like some signal.
So one of the points in this GOLD trial is a combination. That means the lower dose of olaparib. So that may one of the reason any subgroup, even with a patient with DNA repair deficient, did not show the benefit. But I still wonder, this kind of PARP inhibitor may have any impact in overall survival, because this pancreas cancer did not show the benefit in overall survival. So some studies suggested that acquired BRCA mutation as a resistant mechanism. I suspect this may contribute to their shorter overall survival, but maybe we need more data to confirm survival benefit. And another very important target for gastric cancer is the Claudin 80.2.
Yes.
This is a tight junction protein usually expressed in normal stomach cell, but after cancer cell, especially extensive diffuse type show the high expression of claudin. It may be a good target for a monoclonal antibody. And the previous first trial conducted in mainly Germany and Russia showed a good benefit in combination with the first-line chemotherapy. And currently, this was investigated in phase 3 trial. Also, in this ASCO, CAR-T therapy for Claudin 18.2 was also reported. This is a very exciting data.
Yeah, it's interesting. Just like with all of the other tumor types that we have, we're starting to divide up gastric cancer into very specific populations that might have different treatment options, particularly in the research setting. So this has been great. Before we part ways, I'd like to ask each of our panelists for some final thoughts regarding today's discussion. Kohei, give us some final thoughts.
Okay, so yes, recent immunotherapy trial showed some disappointing result, but still may give us some important information. Clearly, gastric cancer is not one disease in terms of response to checkpoint inhibitors. So maybe in the future, we should stratify patients into several subgroups, not only MSI high, but also PD-L1 or other molecules. And small study may give us more insight. And for this kind of a small subgroup, to do a randomized trial in one country is very difficult. So I still believe that drug development for gastric cancer should be done as a global collaboration.
Yeah, very much agree. Manish?
So yeah, I agree with what Kohei said, is that although gastric cancer is a heterogeneous disease, it is and it is prevalent. Drug development is best, in a global setting, because then you can capture that heterogeneity. I think the errors that have been made in the past is that perhaps studies were too small to capture that heterogeneity, and so that may reflect some of the challenges we've had. I think that in the last 10 years, 10,000 patients have actually been enrolled on clinical trials in gastric cancer, and that's very different than even when I started years before that. So there is hope. There are new drugs, and I think the combination of immunotherapy, targeted agents, we're going to redefine how we treat this disease.
David, take us home.
Yeah, I think it's important to stress we now have a global consensus, I think, about treatment approaches. That adjuvant therapy, neoadjuvant therapy works in gastric cancer, certainly a preoperative FLOT in the West, postoperative combination chemotherapy in Asia without radiation, G-junction esophagus, chemoradiation, still the more dominant approach in the West. And I think in terms of new drug development, immunotherapy drugs, at least at the current time, have a role in later line treatment. It's not a clear defined role in early line treatment. And we have a lot of hope for ongoing trials looking at the FGF receptor as well as the claudin molecule, which are very promising signals from phase 2, as well as a whole host of other targets and approaches in development in later line therapy. So. But there is a global consensus, and I agree with Kohei that we need to work together. This is a disease in Asia where trials can be done more efficiently, and I think there's more agreement than disagreement now about how to treat patients.
On behalf of our panel, we thank you for joining us today and hope you found this OncLive Peer Exchange discussion to be useful and informative.